What are you doing up there? Good morning, all. It's lovely being here amid a group that is so emotional about a wonderful job that you all have. And I say this from the standpoint of somebody who's not allowed to be emotional. I'm a forensic scientist. My job is to speak for evidence. While I cheer you on in my heart, if I'm in the courtroom, a different ballgame. I'm up there to present the evidence that you found. I'm up there to present what we could say about it. If the evidence only goes 90% of the way there, that's what we're going to say. We'll say to all of you, we're sorry, please go get more evidence. That's why I'm here today, to encourage you all to hunt down your evidence. Beginnings of police forensic science go back to 1835. At Scotland Yard's Henry Goddard became the first guy to use physical analysis to connect a bullet to a murder weapon. He really didn't, sorta, the markings on the bullet maker, but that's okay, it was a start. There was a flaw in the mold, which he used to make the identification. It wouldn't stand up in court today. 1925, Major Cavalin Goddard established the first laboratory where ballistics, fingerprinting, blood analysis, and trace evidence were all brought together into one facility. He then went on to help design a crucial instrument in our work, which is the comparison microscope. The evolution of police forensic science went from early microscopes, magnifiers, books, to that comparison microscope, which hasn't changed much today, to mass spectrographs, which are so much different, and computer databases, which I barely even want to talk about because I will get emotional. They drive me insane. But yes, I'm a dinosaur. I can barely understand my iPhone. There was an unfortunate early start for wildlife forensics back in the 1950s. A fish and game scientist, and I use that word loosely, used a simple analytical tool called paper chromatography to analyze wildlife blood samples. I'm going to show you how all that works. A little bit of science here. The way it works is you gather these materials together, which in the primary piece of instrumentation is a piece of paper. You draw a line at the base of that piece of paper, about an inch from the bottom. You apply known and unknown blood samples along that line. You drop the paper into a developing tank of solvent that's closed. The solvent rises up the paper. You've seen this happen if you stick a piece of paper in water in the sink. The water rises up. It's a simple technique. And as this is happening, the solvent drags the blood along. And at some point, you stop the process, you take the paper out of the tank, and you realize that your blood stains, assuming that's what they are, have traveled different directions, or the same direction, different lengths. And we're able to calculate what's called an RF value. Sorry about this, but a little bit of math. It's only division. It's the distance that the center of the spot traveled divided by the distance that the solvent traveled. That gives you an RF value. And based on determining these RF values, this scientist determined, one, that the sample he was analyzing was blood. Number two, he determined it was deer blood, again, based on that single RF value. Number three, it was from a white-tailed deer. And number four, he determined that the deer was a female. And every now and then, he was able to determine the grazing area of that animal. This, for lack of a nicer word, is horse poop. Paper chromatography is an analytical tool. It can't do any of what I just described. This fellow, however, testified in court for something in the neighborhood of 7 years. How did he get away with that? Nobody questioned him. He was a scientist. Up there, presumably raised his right hand to tell the truth. I would assume he realized that his science was nonsense. Nonetheless, it went forward. Things have changed. Could he stand up to cross-examination today? No, he'd be laughed out of court or prosecuted, more likely. The real start of wildlife forensics were 6 U.S. state fish and game agencies who had 1 or the most 2 scientists in their laboratory, generally doing blood analysis work. And all they could really do was blood analysis of species within their state. That's all they really had researched and studied. The tools, comparison microscope, octolony diffusion with not very species-specific antisera. If your antisera said it was bear, it could also be raccoon. If it said it was human, it could also be any other primate. Now, the human sense, you're pretty sure there were gorillas running around the city causing havoc, but not necessarily in the woods. The establishment of the U.S. Fish and Wildlife Forensic Science Branch in 1979 started out with Roxy Leyburn. She was my first employee. An interesting lady. The bird lady of the Smithsonian. She had a comparison microscope and the entire Smithsonian bird collection. She also had a 240 ZX. She drove much too fast, as the police frequently told me. Wasn't much I could do about it. So the real start of wildlife forensics, became 10 state agencies in the 1970s and 1980s, to which were added 2 federal laboratories, ours and the National Marine Fisheries Laboratory. Today, we're up to 11 state labs and our 2 federal labs. Canada has joined in. We have laboratories working up there, generally 1 person in a small laboratory area. When it was, Megan said we're the only crime lab in the world, it's not really true. There's lots of small crime labs, 1 or 2 people labs. We're the only full-service crime lab having all the different disciplines of a crime laboratory. Sadly, we would like very much not to be. We're cheering all of your nations on. We will share everything we have if you'll create a similar system. England, the UK, Australia, New Zealand, India, and Southeast Asia. Nothing much in that part of the world. Brazil, 1 person, working part-time, trying to take care of her young daughter while trying to avoid being shot by the poachers. South Africa, Kenya. But going back to '86, there is some irony, and I don't really have an explanation for this, other than I'm part of an interesting Scottish-English family. I started up the first laboratory bringing all these things together. Was Calvin Goddard and the other Goddards my relatives? Yeah, apparently so. I have no understanding how that works. It's probably a flaw in our genetics, but we'll keep it going. To explain a little bit about crime labs, they all do 2 things. They identify evidence in a triangular fashion. We attempt to link suspect, victim, and crime scene together with that evidence. Well, wildlife CSI, pardon the use of the term, for those of you who watch CSI on TV, I'm sorry. I'm partially responsible. I was an advisor. They got it wrong. They know they did, but they made lots of money and they didn't care. But wildlife CSI is very much like the real police CSI, with some differences. Our victim is a non-human animal. There's some 400 police crime labs dealing with 1 species. We get the rest of them. It may be legal to kill that animal depending on a number of interesting things. Its species, you all know about that. The date and time of the kill, before hunting season started at 5 in the morning or after at 7, because hunting season started at 6. Location, 1 side of some country, city, boundary, road, where hunting either is or is not allowed. The type of weapon used. My first case was a golf club on the Congressional Golf Course. The victim was a goose. It honked, apparently, when the doctor was putting. I was in disbelief. I said, "You don't want me going out there and drawing a chalk line around the body, do you?" And they said, "Well, not really, because the goose is gone." I probably should have just walked out, gone back to police work at that point. Possession of a valid hunting license. And the lovely part was its self-defense. Well, if you can imagine all those caveats being handed to a homicide crime scene investigator, they'd lose their mind. You all understand the difference. Species definitions, what we're talking about here, is usually defined by country of origin and by characteristics found on the whole animal. For example, we know this is an elephant. Big ears, large butt, small tail, all the varying things. Everyone in here knows that's an elephant. It's not a rhino. It's not a hippo. Because of those species defining characteristics. We don't get whole elephants in a laboratory, probably fortunately. We get pieces or parts. There's not too many things that look like a elephant trunk ice bucket or an elephant's footstool. But when you get down to the leather goods or the carvings or the tissue, I'm sorry about that, let's go back to the tissue, we completely lost those species defining characteristics. And so what we have to do as a laboratory is conduct a great deal of research to develop new techniques that will allow us to identify species based on their pieces, parts, and products, knowing that those parts and products could come from anywhere on the planet. We cannot make assumptions that just because one of you seized an evidence in some city of your country, that doesn't mean that's where it came from. Your suspect could have been hunting in the United States or in Russia or someplace else. Lab organization. My chief scientist, deputy lab director, and I oversee admin and forensic science. We have 20 scientists in our laboratory, 10 support staff, which makes up roughly 2/3 of the active full-time wildlife forensic scientists on the planet. We would like that not to be true much longer, if you all don't mind. What we're hoping for is at the very least the countries can establish a small wildlife section within their police crime laboratory. We'll share our techniques, our protocols. We need the brains and the hands. Our morphology team, biology, mammals, birds, reptiles, pieces and parts. Using calipers, microscopes, comparison scopes, reference books, crucial things, collections of knowns. We have literally 50,000 known pieces, parts, or whole animals in our collection. Genetics, same basic thing, using DNA techniques to identify species, subspecies, gender, and every now and then we can actually individualize, which is to say we can match a head on a wall to meat in a freezer, blood on hair, car, clothing, weapon. That animal, no other animal statistically in the world, we can do that with about 45 species right now. Genetics team tools, ultrafreezers, PCR techniques, automated robots, which is a little bit amazing to me, having a biochemistry degree from 1968. I had no robots. I also made mistakes, which the robots don't. And sequencers. We have a new tool called genomics. It is Pandora's box, to put it mildly. If I had any brains, I'd pull the plug on that instrument, heave it out the back door, and say, "What genomics instrument?" I'll explain why in a moment. We got this instrument in about a year ago. We ran our first sample. It generated a terabyte of data, linear data. Our computers gagged. I guess that's the proper term for suddenly stopping. We didn't know, we thought we did something wrong, so we sent another sample through. We got another terabyte of data. We don't really know what's in that terabyte of data. It's sort of like having created a Smithsonian library room full of books where we know the alphabet and the definition of a few words. But we do know something. I, for example, know that roughly half my DNA came from mom and roughly half came from dad. Both of them, same directions. We can go out as many generations as you wish, 30, 40, 50. At some point it becomes religious and certainly political. It's going to tell me where in Africa I came from, I'm guessing. The thing is, all that data is in my DNA. We can't read it yet, but it's there. It's going to be readable someday. And is that a problem? Well, yeah, it is. Not for my heredity, I don't care. I obviously evolved to, a less darker-haired person. However that happens, some Scottish thing apparently. But from a wildlife standpoint, it's very much a problem, because if we can look at that wolf sample we ran, and we can go back to, say, the 7th generation, and we find a malamute, does that make that animal a hybrid? Yes, that's the problem. We need to have new definitions of hybrids. We need to have new definitions of species. Politicians, Fish and Wildlife Service, bureaucrats don't want to hear that. Good old scientist. The future, genetic manipulation, it took about, 20,000 years to go from a mammoth mastodon to a modern elephant. We could do the reverse quite quickly now, taking an egg, fetus, or a small calf, and it's almost there. We're going to be able to recreate a mammoth. Or we might screw up, to use a non-scientific term, and create something that's never existed on the planet before and does not have a natural predator. Think a 5-foot diameter black widow spider, for example. Terrifying. Our pathology team, they determine cause of death by conducting necropsies, autopsies on a non-human animal. The tools, digital X-ray, photography, Bluetooth, and strange glasses to use monochromatic laser light sources to look at stuff on the outside of the animal. Chemistry team, probably the most dangerous job in our lab. They identify pesticides and poisons from the necropsy. If there's enough poison in a grizzly bear to kill it, it can knock us off just as easily. We have to be careful. Tools, very complicated mass specs, and other sword instruments. Criminalistics team, primarily firearms, just like it's been done way back when Henry Goddard first started playing with a bullet mold. But things have changed for us because we've been asked to identify planks. When we were first asked that, I thought that was nonsense. We're bunnies and guppies, we're not trees. Turns out we are. If we had a whole tree, same basic issue, leaves, flowers, branches, fruit, seeds, no big problem. But if we're raiding Gibson guitars or seizing planks, it is a problem because we've lost those species-defining characteristics. We'd really like the manufacturers of guitars to leave a little something extra on the soundboard. I don't think they will. But in the meantime, we're making seizures. You folks are making seizures. I'm the scientist. This is what you're seizing. Containerized cargo ships. In one case, 8 of them with 1,000 containers containing multi-million dollars worth of endangered logs. It's tough to identify them there. Once they're turned into planks, milled, and then kiln dried, killing the DNA, we have problems. We have to have a wood standards collection, our woodshed, if you will, which we quickly created, or we're still creating. It's over there on the left. We have cartoons because we're scientists and they make us laugh, and we need that every now and then. Using DART, direct analysis in real time, it's a fascinating instrument. Normally in a mass spec, you have to turn your unknown into a liquid or a gas, inject it. In this case, the DART has a plasma generator, generates a stream of extremely hot helium across a gap. You take your unknown with a pair of forceps, stick it into that gap about 700 degrees Fahrenheit, and a little bit's etched off, and you get your results. And this almost killed my chief scientist. He was analyzing a sample, started to get a headache, tunnel vision, dropped the sample, staggered outside, got medical help. Turned out there's a couple things we didn't know. Number one, we thought all the plasma being generated, the gas, was sucked into the instrument. That's not true. It was billing all around. Number two, we didn't know that rosewood contains a natural insecticide, which is a neurotoxin. My chief scientist basically was shutting off his brain. He had about 10 seconds left. He's better now. Didn't seem to hurt the fact he's smart. We now have a fume hood on our instrument. So he can analyze and he can tell the difference between different types of rosewood. We can actually analyze a plank Now, you determine that it is a species of rosewood. We can often tell where it came from. The science is amazing. We even helped create a app. I didn't know what an app was. I had to ask my granddaughter. She explained. There's now an app out there that you can have in your tablet, which will work a crime scene just the way I would do it, except I wouldn't be using an app. I'm a dinosaur, but it's there. We created a society for wildlife forensic science. There's now about 110 people in that society beyond our 20 scientists. They're research scientists from all over the world. We're hoping to keep adding to them. We're trying to get wildlife forensics going in all directions. We are accredited under ISO accreditation, which, as many of you know, under ISO accreditation, all analytical protocols must link back to long-established ISO standards, such as the standard meter, the standard gram, and the standard liter. The question, of course, is where do we find our standard wolf? ISO had trouble accrediting us. They would like us to have a standard wolf. We tried to explain about a bell curve. They didn't want to hear about it. But we are now accredited as best we can be. So what I'm basically telling you today is wildlife forensics has come a long way from that paper chromatography to genomics and that little DART machine. I don't know where it's going to go into the future. I probably got a year or two left to hang out in this laboratory because it's simply too good of a job to walk away from. But we're here because of all of you. Our job is to support you, to help you do your very dangerous jobs. I wish you all Godspeed. Thank you.
Thank you, Ken. We do have some time for questions. Mark.
You can ask anything. I might answer. Yes, sir. I was digging up bodies out in the desert, San Bernardino, Riverside County, for my first 7 years, and ran a crime lab in Huntington Beach for 7. I'm sorry? I did. We may have crossed paths.
Over here, Miles.
Hear me?
Yeah,
there we go.
There we go.
How do we access your facility?
Through your law enforcement agency. If you're any wildlife investigator around the world, we're the official crime lab for CITES, plus the Wildlife Working Group of Interpol. All you have to do is call us up. Our work is for free. We don't charge. There's a reason. It's kind of like if you drop your car off to a repair shop and said, fix it and give me the bill. You might do that once. You won't do that twice. For us to go through all the evidence you might submit and us to figure out what we could do and what we'd charge, it's too much of overhead. We'll try to do as much as we can, but it is for free.
What if we are already a law enforcement agency and we don't know how to access you? How do we do that?
Well, Google a lab. I think my email address will be handed out. I think I'm easily found.
I found him.
She did.
Was there a question over here?
Okay, my name is Edwin from Kenya. It was interesting to see all that you do, and there was a part which was very interesting about the rosewood. I don't know whether you have the details because you have rosewoods in Kenya, and as well they are endangered, and I never knew about the medicine part which is neurotoxin. Thank you.
You have a nice crime laboratory starting in Kenya. We are cheering your folks on. If you have a wood scientist amongst them, or you can find one, all of our protocols we will turn over to you, no charge, including how things work. When we got accredited, by the way, we had to file all of our protocols. Our application was 4,000 pages. It took us 2 years to put it together, but we got accredited, and I'm looking forward to helping.
We have a question back here, the gentleman in the red. The microphone's coming.
I'm not used to law enforcement officers obeying orders like this and not talking.
Well, we have a translator
Oh,
right
yes,
there,
that's right.
And unless the question gets asked into the microphone, the translator doesn't hear it.
So they have to obey. This is fascinating. Okay.
Just a little personal thing for you. I thought maybe with that big booming voice, after you retire, you could go into radio and television announcing, just to help you out a little bit.
Thank you. I'm not sure I would have the self-restraint on such a show. I'd probably be shot. Aha. He thinks he does remember.
I do remember. But the question is, as far as a prosecutional standpoint, the chain of custody for evidence, what standards does the forest or park ranger need to follow to make sure that what sample they're taking is, held
Properly collected.
Correct.
Contact us first. I mean, you undoubtedly have your own protocols, procedures, which would very likely include chain of custody. We will follow whatever chain of custody materials you send us. We'll also run ours in parallel. We occasionally get evidence from countries where we don't understand the language. We want to make sure there's at least one system we can testify to in court. Contact us. Contact my chief scientist. He'll negotiate with you, and he'll get some work done for you.
Next question? Standing in the back.
My name is Chris Callius from the Game Rangers Association. Just a question around the sort of integration between the human forensics and the other side, and that's the big challenge that we've had in Africa, is that anything related to crime in terms of the human element, that gets processed by a different forensic laboratory. By the time they've finished with it, the ability to access any forensics to understand the natural features of it, so the species, etc., has all been removed. So the challenge is about integrating that so that we have right at source the right collection. And I think the challenge is in other African states is also accessing that, so capturing samples and then getting those samples out of the country is a whole nother thing, challenge in the system.
Yes, bureaucratic paperwork is interesting. There is no exception in CITES for law enforcement, as some of you undoubtedly know. You'll have to get a CITES permit to send something to us, which is an interesting bureaucratic morass. I'm being nice. But contact us. We have two senior special agents, liaison agents, one in Tanzania, one in Gaborone. They're contactable, or you'll have my email address. Get a hold of me. We will make something work. I've been to several countries in Africa, and I know some of the laboratories. They're fascinated by what you all do. Let's get them fascinated with the science as well.